Well, good evening, everybody. I'm J.D. Hansen. I'm the Policy Director at the Center for Food Safety. This webinar on genetically engineered mosquitoes is co-hosted by the Center for Food Safety. I want to thank Maria Ure from our communication staff for making sure that everything works right. Dana Pearls, would you introduce yourself from Friends of the Earth, one of our other sponsors?
Hi, my name is Dana Pearls. I'm the program manager of the Food and Technology program at Friends of the Earth. Thank you for joining us for this call.
The Florida Keys Environmental Coalition is the third of our three sponsors, and Barry Ray is the executive director. Barry, would you welcome the group?
Hi, I'm the executive director of the Florida Keys Environmental Coalition. I'm one of the locals down here. I'm pleased to have you all join us. This is a very important issue, and we welcome your questions and really look forward to your participation. Thank you.
Let me explain some of the procedure we'll be using. If you have, in the next 10 minutes or so, some technical problems, use the chat function for that. After that, the chat will be turned off. If you have questions for the panelists, Barry and Dana will be going through them, and we will answer them at the end. If there's something that you just had a burning question and didn't understand something, put that in the question, and we might be able to get to it while the presenter's still up. Please put your name, and if you're a member of the Center for Food Safety, put, J.D. Hansen, Center for Food Safety, ask a question. If you're from the Florida Keys Environmental Coalition, put that with your name so we know who you are. If you do something anonymous, it just won't get answered. Now, our first speaker is Dr. Jeffrey Powell, who is an entomologist at Yale University, and he told me as we were starting this that he has been studying mosquitoes for 50 years. So he knows mosquitoes. He assures me he didn't actually know Rachel Carson, but he's been studying what she learned as an entomologist all of these years, and I don't want to cut into his time. Dr. Powell, welcome.
Thank you very much. I know I don't have much time, so I'm going to go over this pretty quickly. And this has to do with a release program that was carried out in Brazil, in a town called Jacobina, Brazil. And this particular release program was designed so that male mosquitoes, male Aedes aegypti, were released, and they're carrying a lethal mutation so that when they mate with females in the wild, there's this right side of this little cartoon shows you here, the larvae get formed, but they die before they become pupae. And on the left side is just the technical molecular biology behind this. We don't have to worry about this. And so that's the idea. You release a lot of these males, you release enough of them that you suppress the population, because the females they mate with should not produce offspring, or very, very few offspring. And we'll get to that in a minute. In the next slide, we'll just show you where we work in, and this is a place called Jacobina in the state of Bahia in Brazil. On the lower left there, you can see where Jacobina is, and then there's an aerial photograph of the city. It has different neighborhoods. It's about 3 miles across from the upper left to the lower right. And my co-author in this, Ben Evans, is a graduate student who participated in this, and he's showing they're releasing some of these males. The released males, let's have the next slide, were produced in a place in Juazeiro, Brazil called Moscamed. And this is just a photograph of the workers in Brazil producing these millions, hundreds of thousands and millions of mosquitoes. And I think it's important to note that this is expensive from a labor, it's highly labor intensive. And I think this program probably had more than 20 salaried workers to produce all of this thing, all of the mosquitoes needed to carry out this program. So this program was carried out, as I say, in Jacobina, a city of about 50,000 people. And the next slide, I think, okay, the next slide gets a little bit complicated, but let's not worry about it too much. The little graph in the upper right is just there to show you that our genetic technology, the purple dots in that upper right little graph there, are the release strain. This was called Oxitec, Oxy 103A, I believe is the name of the strain. And on the right there, those dots are the populations from the release in Jacobina before releases. The point of this is that the Oxitec strain is genetically very distinct. There's no genetic overlap between the Oxitec strain and the population in Jacobina before release. So we can really distinguish them. And the next, yeah, let's go to this one. And so what this graph is, it gets again a little bit technical, but I think all you need to really see here is that on the far left here, each of these little lines that are going up and down, there's many hundreds of lines going up and down. Each of those lines represents a single mosquito that we genotyped, okay? And on the far left, that solid red is a bunch of the release strains that we genotyped, and we've indicated them as being 100% red. That should go all the way up to 100% red. The F1 are fluorescent larvae, which would mean that those are offspring from the release males and the wild type females in the Jacobina population, and they should be about 50% red and 50% blue, as you see. The next blue, the solid, go back please, I'm sorry. The next two blue blocks there are Jacobina natural population of mosquitoes before releases began. Okay, and so clearly you can distinguish the release strain, which is red, and the blue, which is Jacobina before releases began. And then as we move to the right in this graph, you see the different neighborhoods of Jacobina, Centro, Catuaba, Pedro Branca, et cetera. You can see after 6 months, 12 months, and we've followed this for 27 months after releases started, you begin to see some of the Oxitec genes, ancestry, or what do you want to call it, from the release strain are now getting into the natural population, the population in Jacobina. And that's what these partly red, partly blue individuals are. They're mixed individuals between the release strain and the natural Jacobina population. So genes are getting transferred into this population. And so the next slide just says, okay, so we published this, and this is just the first page of our publication. It caused some stir, and the editors of the journal, which is Scientific Research Journal, published under pressure, published what they called, the next slide, what they called an editorial expression of concern. The first thing I want to point out before I address that is that no one has actually challenged the scientific validity of our study. No one has challenged the data. No one has challenged the conclusion that genes have been transferred from the release strain into the natural target population. That's solid, and we stand behind that 100%. This editorial expression of concern that the editors decided to do had to do more with the wording, I guess, is what their problems were. That it was not objective, that the language in the paper is alarmist, there's ambiguity and speculation that cannot be justified. I disagree totally with that. I don't believe that at all. So I just thought I'd say, what did we actually say in the paper that could have caused this kind of concern by the editors? So the next slide will just show you, let's see. Okay, no, go, okay, here. Okay, so one thing that we've been accused of is that we overinterpreted it and we said that this was really bad and was going to have problems because of disease transmission, et cetera. This is not true. I can quote from the paper itself. Here are two sentences that come from the publication. In the abstract, which is a summary, we say it is unclear how this may affect disease transmission or other efforts to control these diseases. We repeat that again in the discussion. We point out honestly that it is not known what the impacts of introgression from a transgenic strain Aegyptia has on traits of importance to disease control and transmission. We say that in the paper. We do have one sentence, which I think is the one that probably caused some concern. And that's the last sentence here, is that in fact, the three populations, because the release strain was a mixture of Cuba and Mexico, and so that Cuba and Mexico release strain was now mixing with a Brazil strain, so it's now a three-way hybrid population. And we just say that it very likely resulted in a more or less robust population due to hybrid vigor. Hybrid vigor is very well known in genetics. When you cross two genetically distinct strains of corn or anything else, the offspring have more vigor than the two strains begin with. So these are the sentences that we actually have in the paper. And my, I personally do not see what's alarmist about these kinds of things. And so this is what we've been, and what the this editorial concern was about. And personally, I don't see anything alarmist in this, and I don't want to belabor this anymore. I do want to make one other point before, and I think this is important. If I could have the next slide. One thing which has been kind of overlooked in this release program in Jacobina that went on for more than 2 years, 27 months, and that is that the effectiveness of this control program began to break down after about 18 months. Okay, and so the next slide will just show you the data, and this is again a little bit sloppy, and it's, I apologize for it, but these are the data that come from the people who are doing the release. What's important here is to look at the green thing. So here we have the releases began in 2013 and went on to the end of 2015. The blue thing shows a number of males being released, which is over 500,000 males per week were being released. But what's important, what I wanted to point out here is this green line. The green line is what's called the ovitrap index. And the ovitrap index is a measure of the female adult population size. And so what you see is there's a lot of fluctuation, but before the releases began, the population size was up here on average. As the releases started, it was working. This green line went down. It seemed to be going down quite well for about 18 months. And at about 18 months, the green line goes back up. So the next slide just shows this in a bit more. If I could have the next slide. Again, these are the same data. Now you can see it with the black line, or that same data, or the ovitrap index, measure of the adult population size. Before releases, it was high. Releases started, this started going down. In about 18 months, it goes back up. So something happened around 18 months that the program to suppress the population size started to fail. And what the evidence is, and I won't go again into the technical aspect, the evidence would point out that it's quite likely that the population has been selected to discriminate against mating with the release strain. This is well known in sterile male releases of insects after a certain number of generations. And this time, in this case, it looks like about 18 months of weekly releases of 500,000 males. The selection against mating with them worked, and now the females are no longer mating with the release ones, and so the population came back. So I think that's another important aspect of the technology being employed here. So I think, let me go on to the next slide. Okay, so I'll wrap it up here now. I think my time is just about up. I want to first of all point out that I am not anti-GMO. I don't have any philosophical or ethical problems with genetically modified organisms. I eat them every day. I think there's a lot of potential of genetic modification of mosquitoes for controlling mosquito-borne diseases. We have some ideas we're working on, but I think what I would point out is that the approaches to date, I think, have some problems, and the same approaches are being now proposed in Florida. And I would just point out the 3 points I would make about this is that from our work, it does seem like genes from the genetically modified release strain get into the natural population, or what we call introgression, with unknown consequences. And I think I cannot claim I know what the consequences are. But it's also important, I think, to show, to understand in advance that the effectiveness of the suppression will likely break down. And again, this is well known in these kinds of sterile male releases, when your releases are, in a sense, the same kind of thing, but it's technically somewhat different. And the final point, as I pointed out earlier at the beginning, was that these methods are labor intensive, and expensive, and need to be continuously implemented. As soon as you stop this kind of control program, the population will come right back to where it was. And so I think there are problems with what is being proposed. But finally, my very last slide and the take-home message from our study is, this is a very final sentence in our publication, and that is that we demonstrate the importance of having in place a genetic monitoring program during releases of transgenic organisms to detect unanticipated consequences. So if there's anything that our study, I think, shows, is the importance of having this kind of monitoring program during any releases of transgenic organisms. And with that, I will stop.
All right, thank you. Thank you very much, Dr. Powell. We have a few questions, but I think Dr. Helen Wallace will cover some of the same territory, so we'll save the questions we have to the end. I would welcome folks that have come in later. We've had a few additional folks join us. I urge you to please put your name and organization. If you don't have an organization, put where you live. We won't take anonymous questions. That's just a policy we've made. Now I would like to not cut into Dr. Helen Wallace's time anymore. Dr. Wallace is the director of GeneWatch UK. They have been the organization monitoring the development of all sorts of genetically engineered insects for a number of years. And I'm glad that Dr. Wallace can join us, even though it's quite late in the UK right now. Dr. Wallace, please.
Thanks, JD. I'm really pleased to be here. Could I have the next slide, please? Yeah, so we've been following Oxitec's work since about 2009. And I just wanted to start with some numbers here in the right-hand column, showing you the numbers that are planned to be released in these experimental releases in Florida and Texas. And you can see that even for these quite small-scale experiments, you're getting up to hundreds of millions of genetically engineered mosquitoes. And the idea is to release the male genetically engineered GE mosquitoes to mate with wild females. They're not sterile. What they do is the wild females will lay eggs which contain the genetic changes, which include the killing mechanism and the fluorescent mechanism. And in all the experiments to date, the ones that Professor Powell was talking about, for example, both the male and female offspring were supposed to die at the larval stage. But this is a new one that they want to trial in Florida. It has been in a trial in Brazil, but there's not been any published data about it yet. And in this particular iteration, if you like, the second generation, Oxitec calls it, only the female offspring are intended to die. So that's basically how it works. Can I have the next slide? So first of all, the issues about efficacy. Again, Professor Powell has already talked about how the effectiveness can wear off over time as the mosquitoes adapt. This is just a small part of the research we've done, highlighting that Oxitec's claims of efficacy are not backed up by the data that they actually have. So these comments that I put here in italics were made by scientists from the Cayman Islands Mosquito Research and Control Unit, MRCU. And you can see they're saying that whilst Oxitec and MRCU are making public statements complaining major reductions in the Aedes aegypti, that's the mosquito species, the data I have seen does not support this. And to date, all the measures recorded have shown no significant reduction. So these are comments from the scientists who could actually see that data. And also they saw a significant increase in the number of the female mosquitoes. Those are the mosquitoes which bite and spread disease. And that's probably because female mosquitoes were being inadvertently released during the experiment. So do take a look at that briefing if you want to learn more. Next slide, please. Well, why is efficacy important before I talk about risks? Well, it's really important to know this new version of the which is called the OX5034, that's the one they want to release in Florida and Texas, is female killing only. So instead of both males and females dying at the larval stage, only the females are expected to die at the larval stage. And that means you basically have twice as many mosquitoes in each generation as you did in Professor Powell's experiments. So you can see here, I've numbered all the EPA documents, but the EPA itself is saying the degree of introgression, that's the spread of genes that you've just heard about, is likely to be significantly higher than in the earlier OX513A strain that you've just heard about. So the highest larval survival rates differ. They're not just around 5%, but 50%. So expect these genes to spread a lot more into the wild population. And a key issue with that is whether those genes come from a strain of mosquito, which was taken from Mexico originally, that could spread the disease more rapidly. So we just don't know, because no studies have been done, whether the vector competence, the ability to spread that disease of this, what Oxitec's proposing to release in the environment would actually be worse in terms of spreading diseases. Could I have the next slide, please? Will the genetically engineered female survive in the environment? Well, there's two ways that could happen. One is that they could evolve resistance to the killing mechanism. So they want to survive. Random changes in their genes might allow a few to survive, and then they can evolve resistance. The EPA says it hasn't considered that yet. It's going to think about it later. Should it receive a request for registration, then it will consider whether a program designed to manage the potential resistance is feasible and warranted. So we don't know whether that's going to happen. Another thing you're probably going to hear more about later is that the antibiotic tetracycline is used like a kind of an antidote to the genetic killing mechanism. So Oxitec need to have females surviving to adulthood in the lab so that they can breed them. So they've built in this kind of antidote. And tetracycline exists in the environment. And we've found evidence, which Oxitec later admitted, that some of its genetically engineered mosquitoes of the earlier version actually survived. 15 to 18% of them survived when they were fed on cat food, because the chicken probably had sufficient tetracycline in it to increase that survival rate. Now, in the documents that the EPA has released, they're suggesting a couple of safeguards. They're going to keep the releases away from sewage treatment plants where there might be tetracycline, and away from commercial citrus growing where tetracycline can also be used. But is that going to be enough to stop the female survive? Again, we just don't know. Could we have the next slide? Why is this important? Well, is it safe for humans? Is it safe for animals? First of all, you need to think about the direct effects. And again, the new documents from the EPA say no determination of the allergic or toxic potential for either of these two new proteins that are genetically engineered into these mosquitoes has been made at this time. So they say they don't know if it's safe in terms of allergy or toxicity, but they're assuming that people won't be exposed to them. But they could be exposed if anything goes wrong, as I explained in the previous slide. So if any females can survive, and they could also be exposed if you swallow them. While swallowing mosquitoes, is that very likely? Let's have a look at the next slide. Again from Brazil, this is a report from the LA Times about some of the experiments in Brazil, where they said it's impossible to talk during the liberation sessions without accidentally swallowing a few. How come? Well, because Oxitec was releasing 54 genetically engineered male mosquitoes for every one wild one, because they needed that huge number to see any effect on the wild population. In addition, don't forget that the female GE mosquitoes will be dying at the larval stage, so they'll be dying where they breed in little pools of water. Now, it's more of an issue in poorer countries where people might be drinking that water. You're probably drinking tap water. There's still an issue for animals. And Oxitec plans to release, as I showed in the first slide, hundreds of millions of these GE mosquitoes. And that's just for the experiments. If it ever gets commercialized, the numbers will be a lot larger. Could I have the next slide, please? Indirect effects can occur if the population goes up, then down again. You can actually have effects on other mosquito species. So one particular concern is if you reduce the Aedes aegypti mosquito, which is the target one, even temporarily, could a different species called Aedes albopictus, which also transmits disease, move into that ecological niche. And Oxitec itself have admitted, yes, that possibly could happen, but there's nothing in the risk assessment about that from the EPA. Next slide, please. Will it reduce spraying? No. Partly because Aedes aegypti are only a small proportion of all the mosquitoes you have there in Florida, but also in the EPA documents themselves, they say they're going to use exactly the same treatments as they did before while they do the experiment. And that's a potential problem as well, because they could be killing the supposedly good mosquitoes as they try to release them. Next slide. So, sorry, that's a repeat of what I was going to say about efficacy. The efficacy is not going to be tested by these experiments. Why? Well, the mosquitoes that are being released are not going to be tested for the direct influence on disease transmission, whether they actually make dengue or Zika any better. And that's agreed between the EPA and Oxitec. But Oxitec is even saying it shouldn't have to show that it can suppress the wild population. It's saying that it's only going to look at whether the offspring of the mosquitoes that mate actually die more than the wild mosquitoes. And the final point, discussions are ongoing with the EPA. Fully informed consent, while you've not been fully informed, all the documents I've talked about were released after the consultation, dozens of pages of information, and fully informed consent is not required under the EPA's human studies rule, according to the EPA. Final slide, please. So to sum up, Oxitec has made misleading claims about its GE mosquitoes in the past, particularly in relation to efficacy, but also in relation to their spread into the environment. It's not tested the strain that it's going to be released, which originally comes from Mexico, to see if it could transmit diseases more than your local mosquitoes. It's not provided sufficient evidence to show that it's safe. It's not assessed any complex environmental effects, such as whether Aedes albopictus could move in. And its experiments won't show whether or not this works or not. They don't reduce the need for spraying, and there's been inadequate consultation and a lack of fully informed consent.
Thank you very much, Dr. Wallace. We've received about a dozen good questions, but I think there's enough overlap in them that we can wait until the end. There's some questions for you and some questions for Dr. Powell. And now I would like to introduce Dr. Jack Morris, who is a physician in the Keys and has developed a physician's petition around the issue of bacteria. This is one of the many issues that all of our organizations care about, and I won't cut into Jack's time anymore. Thank you, Jack.
Okay, 8 minutes. I'm Dr. Jack Norris, Chief of Staff at Lower Keys Medical Center, the largest hospital in the Florida Keys. We were the hospital whose catchment area involved the initial version 1 release plan for the GM mosquito, OX503A. Basically, our concern was, first of all, you have to understand, we physicians love technology. We're technology lovers. We use GM products constantly, and but we read the small print. We're such avid users, we actually look at what it is that we're being asked to use. I am one of the physicians who fought dengue back in 2009 and 10, and now we have dengue in the Northern Keys having resurfaced itself. Well, on August 17, 2016, the physicians in the Keys signed a petition that I wrote showing great respect to our mosquito control personnel. We asked, because the nature of this technology uses an antibiotic, a chemical called tetracycline, which is classified as a very important antibiotic to human health. It uses it as a doorstop, and a lot of physicians will look at that as trying to take something like a AK-47 military rifle and using it as a doorstop. It's not practical, nor is it wise. So what we asked was not that the project be scrapped. We asked that culture the adult to be released mosquito, this GM, and see if by going ahead and making it a mosquito dependent on an antibiotic as a antidote, as was stated earlier, to a death gene that has been implanted in every cell in a mosquito. Well, what did you do to its bacteria? And I'll explain that more in a moment. Will you propagate resistant bacteria that will cause resistant infections in humans and animals? We ask that they not only do the culture, but factor this into any release and study it. Next slide. Now, we know that mosquitoes kill 725,000 people a year, more than humans, more than snakes, more than all these other creatures. Next slide. We've confirmed dengue is back in the Northern Keys. So we have an eager need to try and treat this vector. Next slide. Females are the ones that transmit the viruses. When they take their feedings, that's when we're at risk. Next slide. The way that this has been done is DNA has been taken from other sources. A death gene and a marker gene have been placed on a small strand and inserted into a mosquito egg, as demonstrated here. And you can notice at the bottom of my slides, I give you specific references if you wish to go and look for yourself at what is present on those pages, where we got this information. Next slide. Once this is incorporated into the egg, next slide, babies are born. So when a GM male mates with a wild type female, all of the children should have DNA from both. Well, the gene being passed by dad is the lethal gene with the marker. So this demonstrates that, again, with reference. Next page. The antidote looks like this, according to the Brazilian group that, that documented their process. Next slide. And you can see these are maturation trays, as that's what I call them, stacks of them, where the antibiotic is being infused into the trays. And of course, look how the tray is empty right now. It has no larva, it has nothing in it but water. Next slide. This is an actual PR picture from the Cayman News. What it shows is it shows a gentleman on the right in the left pane. That is Dr. Petrie. He is right now the head of Miami Mosquito Control. Next to him is an Oxitec employee working in the Cayman Islands. At this time, Dr. Petrie was running the MRCU, the mosquito control in the Caymans, and he led the project. And you can see them inside of their mosquito factory. The concern I had is immediately you go look at the floor and they spilled some of that water that has tetracycline onto the floor. We physicians get very concerned about resistant bacteria. As a matter of fact, one of my old jobs was I used to run the performance improvement of what is now the Rutgers, New Jersey Medical School's ambulatory care for a large section of its ambulatory care. One of the biggest things we cared about was not propagating resistant infections. Well, if you look at it, their shoes have this antibiotic fluid with whatever bacteria all over it. For any bacteria that is in that water, and we all know there's bacteria in water, the bacteria had to survive that antibiotic in order to be alive on their shoes. And then they could track that all over. Next slide. This is what the draws actually look like according to the Cayman News Service again. Kind of looks like sewer water. The picture doesn't show well on the lower right the amount of dirt on the inside, but what you can see is that they're making hand contact, so they're exchanging bacteria with the draw. The draw is full of this kind of brown-yellow fluid, which goes along completely with what we saw for the antibiotic antidote. So again, as far as the bacterial control in this site, it is extremely concerning. Next slide. We know that every living organism, every large animal such as us has germs. We know that cockroaches have germs, flies have germs. Well, mosquitoes have them too, just no one ever really worried about physical contact from a mosquito because it's in our same environment. Next slide. This is an example of 2 Petri dishes with the same type of bacteria on them. Notice the little white circles, those are a filter paper with different antibiotics. The Petri dish on the left shows what a sensitive, what a non-resistant bacteria does when you put these antibiotics upon it. Notice the large areas of death. The bacteria cannot survive in those large clear areas because the antibiotic kills them. These are not resistant germs. The question that we as physicians asked in the petition is whether or not by going ahead and releasing a antibiotic-dependent, genetically modified, man-made species, did you find a way of making mosquitoes have a new health problem for us? If you look at the right, you see where some antibiotics, the 2 lower white circles, had no effect on the bacteria. They kind of ignored it. A couple others had tiny effects, and only 2 still had normal effect. Next slide. All right, now how does bacteria transfer? Physical contact. This is actually a picture from a television show where a person goes to pick up a tissue on the ground in a hospital, throws it in the garbage, and you notice everything he touches is supposed to be contaminated in green with bacteria. He throws it in the garbage, he shakes the woman's hand. Next slide. She touches her other hand, she touches her face. Notice how quickly she contaminates herself. This kind of reminds us what we're watching on television with coronavirus, isn't it? It's very easy to go ahead and transmit infectious disease in the way that we're describing. Next slide. So what exactly happens when you mass expose a insect that has its own bacteria in a fluid by the thousands that looks the way that fluid did, using an antibiotic in the doses that can soak into every cell in that insect's body, get into every cell, and shut off a genetic switch in its DNA? That was the question that was being asked. Next page. This is what it looks like version 1 when they released it in Brazil. Notice the small little dots are the mosquitoes being basically let out of the truck from that was deploying them. Notice how dirty that round exit point is. What kind of bacteria are sitting there? I would love to culture that, because in a hospital setting, we would lose our minds seeing that amount of debris sitting on anything. It is just so unclean. And again, these are antibiotic affected bacteria. Next slide. So the mosquito control held a workshop back in 2018. And you notice the gentleman standing at the podium is Dr. Petrie, who I introduced you to earlier, who's standing in the puddle. And I had to apologize to Dr. Petrie because he attended this meeting and I had shown that picture. And he is truly a consummate scientist, and I have great respect for the man. It's just that, you know, we physicians think about antibiotics a lot more than entomologists do, apparently. And his statement was very clear and can be played if people would like it to be played, because I gave it to the moderator. You don't, oh, you're going to play it? Play just 20 seconds. We're not hearing it. Is there a way of raising the volume? Volumes looks like it's, go back
sorry,
down.
yeah, I'm so sorry to interrupt. No, we cannot turn up the volume. I'm so sorry.
No problem. Pass on it. Just leave it posted for people who want to hear him speak. I put it on my Facebook page, guys. So, all right, next slide. What he stated was that you cannot use genetically, you can't use sterile insect techniques in a disease situation like what we have in Key Largo. The statement was that it's a strategic tool, and he explains more, and the video explains it. So I'm hoping that it is posted easily for you. But if you go to John W. Norris, MD, the Facebook page, you can see the video and play it. I actually flew down to the Cayman Islands. The Cayman Islands had these editorials in their press, the revealing and disturbing MRCU Oxitec emails. I came down, and you see me standing there. To my left, you have my son, Matthew, who's now a physician in New Jersey, treating his own COVID patients. But I brought him down to show him how you chase when you want to find out what's wrong with your patients or what your patients are at risk of, how you go and physically go find the data, because that's what I want here is data. The man to my son's left is the then head scientist, Dr. Wheeler. Dr. Wheeler is a very, very great Cayman patriot who cares greatly about mosquito control. And you're about to see some more on his emails, which Dr. Wallace mentioned earlier. Next page. This is an email that was released by a Freedom of Information Act request. What it says is, it's from Alan Wheeler. It says, "Good morning, Nancy," who is the director at the time. "I do not agree with the responses we are giving to this journalist. I do not agree with a 62% reduction claim. None of the scientists in the Mosquito Research and Control Unit would have said the project was a success. Even Dr. Petrie picked his words carefully when he stated he believed the technique could be made to work." And at the very bottom of the page, to skip down, you can see before this, before I send this up to ministry for final approval, please can you advise if this is okay by Oxitec? You know, which still confuses me to this day why that should be important, but it gives you the date, and this is one of the FOIA released emails. Another one stated that the production facility had a problem with mold. Well, remember, if a person takes an antibiotic, what do they get? They not only do we get concerned about resistant germs, but fungal infections, yeast infections, mold. Next page.
Dr. Norris, are you wrapping it up now?
Now, sir.
Okay.
So version 2. Version 2, basically what they want to do is they want to take these egg boxes, like the lower purple area there, and they're stating that they want to release these, they'll put these boxes on people's properties. Next page. There was statements that this is not an experiment. This is a printout from regulations.gov. The summary states that it is an experimental use protocol. I highlighted that, and tetracycline is in use. So this is an experiment being done using people's homes. Next page. They express that there's going to be approximately 350 boxes. A lot of this has been explained before. Next page. And now look at the grid. Let's go to the lower pane since time is short. Notice they have all these little circles where these boxes go. Those are going to represent people's homes, because remember, the Aedes aegypti mosquito is on us. In order to go ahead, it wants to feed on us, so it lives with us. So they have to put these things where people live. So my question during the most recent workshop was, why wouldn't people, considering that physicians are asking for information that the company is saying they cannot give us, but the EPA said, yeah, they can do it. They just have to give us the information too. Why wouldn't people just go get a plastic bag, grab this egg box off their property and throw it in the trash? And how does that help them do their science? Next page. Approvals have failings. This is just a quick 32% approvals from the FDA. Had big safety concerns post-approval. Next page. We're wrapping up now. So again, the proposal is please test your bugs and factor it in. Next page. Last but not least, Alexander Fleming, the 1945 Nobel Prize speech that he gave, he got the Nobel Prize for antibiotics, said, then there is the danger the ignorant man may easily underdose himself, and by exposing his microbes to non-lethal quantities of the drug, make them resistant. Who is responsible for the victims of the ignorant man? Or who is responsible for the victims of the misuser of antibiotics? Next page. And again, that's the question physicians want answered. And EPA says, Oxitec, you can do it. Mosquito control, you guys are the ones who make the contract and the agreement. Why don't we have this information? We measure our physicians by the dozens in the keys. You've got dozens of signatures. All right, I thank you.
Thank you very much, Jack. We'll have some questions for everyone in just a little bit. Ed Russo is the president of the Florida Keys Environmental Coalition. Ed, thank you for co-hosting this with us. And what should we do now?
After listening to all those reports, I'm not sure exactly what to do, but here we are. I'd like to thank everybody for joining this webinar. It's clear that we have some challenges before us. So yes, I am a local in the Florida Keys. I have some things I need to talk about. First are local concerns, but they're not site-specific concerns. They're concerns that should be addressed, whether it's the Florida Keys or Texas, and any other place that Oxitec attempts this venture. Also, I'm going to talk about EPA and what their personality is right now and what's been happening in the EPA. Obviously, the EPA should change their name to the CFD, the Center for Deregulation, because as you know, that's all they're doing. It's like a big shark, and instead of eating things, it just eats regulation. So it's important everybody to realize that their approval has to be looked at carefully, and we hope that the Mosquito Control Board holds the EPA's foot to the fire, and Oxitec ask them the questions that are coming up today, because you're elected officials, and you should be making sure that they have answers to all the presenters today, especially the questions that were brought up by Dr. Norris. Also, we want to bring up, talk about the false claims. And lastly, we want to talk about who benefits. And it's not us. As you will find out, the only people that are really going to be benefiting from all of this because of the ineffectiveness of this particular technology are the stockholders of Oxitec. So let's talk about the concerns. Our concerns grew when in the beginning we were asking some very basic questions. We weren't in opposition of genetically modified anything. We just wanted to make sure that Oxitec knew what they were doing, and there was appropriate oversight by the citizen group, our mosquito control board that was elected to oversee this. Unfortunately, the mosquito control board really hasn't brought up any standards or guidelines or restrictions. Again, you have a situation where these mosquitoes now are going to be released in hundreds of millions without any controls in terms of the environment or location. Again, they're talking about some of them, but in terms of proximity to schools, churches, hospitals, and senior citizen centers, we don't see any restrictions there or environmental considerations. We've been asking about this repeatedly for years, and whether there is a limitation of wind direction or wind speed, it's important. We have birds blown over here from Cuba. You can imagine how far these mosquitoes can go in the wrong wind conditions. So it's important that our concerns in terms of making sure that we understand the regulatory requirements for release is more important. That, again, we're wanting to ask the question so we make sure that Oxitec knows what these limitations are and these restrictions are, and that the Mosquito Control Board is going to oversee it properly. Unfortunately, the feeling is in the Florida Keys is that our Mosquito Control Board hasn't held Oxitec to any of these standards, and therefore appears merely to be a rubber stamp for cheerleaders. And that goes for our media also. The question is, how can you guarantee there's no females going to be released? That's been the contention. That has been the fundamental building block of all of this. Because if females are going to be released, this whole charade collapses. And so we have kept on asking that question. And we have representatives from Oxitec and the Mosquito Control Board on record saying there won't be any. This is the first time they wanted to bring this out. And that turned out to be absolutely false. That after information that was gathered by our people, we found out that somewhere between 8 and 18% of the mosquitoes that were going to be released were going to be females. What could go wrong with that? My God, these are important issues that have to be addressed. And so we would like our representatives in the media, we certainly would like members of the Mosquito Control Board to focus on that particular question. How can they guarantee, or can they guarantee, that no females, not a very small, not a de minimis, no, none. Because if you get a few females out and they mate and they have a hatch and they can do that, and again, females can only survive by drinking the blood of humans. Well, now what are we going to do? So we want to make sure that if they say that only males are going to be released, that we want to make sure that that's correct. So we want proof of that, not just technobabble. We want to make sure that when questions are asked tomorrow afternoon or anytime during this entire process from the people from Oxitec, that the Mosquito Control Board holds them accountable. And when we don't see that, and in order to promote public support and public policy, there has to be accountability, and we just don't see that accountability. So that's when it comes to our concerns. Answer specific questions. Our response when we ask these questions, same questions we asked in 2012, is that, well, if we answer those questions, all you're going to do is ask more questions. That's not a way to create support for a particular public policy. Answer the questions, satisfy local concerns. So that's the problem we have with local concerns, that we haven't had our questions answered and or haven't had the forum to discuss this. The EPA is a whole different situation. Again, it just seems odd that the same people that have been railing against this particular EPA of being not effective in terms of protecting the environment are okay with this. Makes no sense. The EPA is on a mission to deregulate everything it touches. We can't rely upon our EPA to protect us. We're relying upon our Mosquito Control Board to protect us. So what has happened is that the EPA, the State Department of Agriculture, and the FWC and the Mosquito Control Board are all depending on the information submitted by Oxitec that was developed in Brazil. Please. You know, then we find out that, well, their statement that this has been tested all over the world is a bit disingenuous, because you have it being tested in the Caymans, and you heard about that. Just so you know, Dr. Petrie is now responsible for mosquito control in Miami-Dade, a huge job. And it's important to recognize that he does not want this to happen. There's no, I mean, he is not supporting the release in Miami-Dade, and he says it's not going to have any effect on any kind of outbreak of dengue. So why are we doing this? It's a very expensive proposition, has to continue forever. So there are other ways of doing it. Again, our interest has always been to promote Wolbachia, which has been tested in Florida Keys, and the Mosquito Control Board has been very careful not to talk about it very much. We're very interested in the success of that test. Excuse me. So the EPA's approval of this has to be couched in terms of what the testing was on. There has been no biological or clinical tests. They have never taken female mosquitoes that could be released and have them bite volunteers, which is normally how they do these things, and see what the results are, or release these mosquitoes that can be eaten or consumed in some way to our endangered species or any species in the Florida Keys or Texas, and look at the results of that. So they haven't been any clinical or biological tests on this particular mosquito. Excuse me. And so when the FDA originally said that this is okay, or the EPA says it's okay, or Brazil says it's okay, or Oxitec says it's okay, what they're talking about is, the most important thing is they're only talking about tests on the mosquitoes, not on human beings, not on the food we eat, none of that. And lastly, I just want to talk about how ineffective this is, how much this will cost, and what it will do to reduce disease, not just the Aedes aegypti, but the diseases that are involved in this. And that analysis has never been created. So let me end by saying, again, the only people that are going to be benefited by this are the stockholders of Oxitec. Thank you.
Well, thank you very much. We have a number of questions to answer our panelists. I'm going to, there we are. Thank you all for joining us tonight. We're going to go about 15 to 20 minutes more. You know, I would note that all three sponsoring organizations have given the EPA notice that we're filing a lawsuit because the EPA did not do the required endangered species review on these mosquitoes, and that did not consult with other agencies like Fish and Wildlife Service and the National Marine Fisheries Service, which is a requirement under the Endangered Species Act. So in a little over a month, we will be suing them. There is a requirement in the law that we give them 60 days notice, and we've given them that 60 days. Now, for your questions. Excuse me. This is a question for Helen Wallace. So Helen will need to turn on her video and that. The question is, will robust hybrids contribute to the evolution of more aggressive mosquitoes and the spread of new diseases? Are there public data that relate to this? And I actually think that Dr. Powell might want to say something about this too.
I suspect that Professor Powell and I have a similar view. Whether we don't know, but there is a risk that that could happen. So we don't know whether the Mexican strain that's going to be released, and whether when it mates with the wild mosquitoes and spreads its genes in the population, whether that is going to alter the properties of the wild mosquitoes or not. So we can't answer that question, and there is no data, and there are no plans to collect any data at the moment.
I would just modify that by saying, you know, in genetics, there's a long history of demonstration that the more genetic variability a population has, the more robust it is, the faster it can evolve. And that's why, for example, almost all the crops that one plants are hybrid seeds, because the hybrid is more robust. And so I would just argue that this idea that when you hybridize between genetically distinct populations, be they a plant, a mosquito, or whatever, the offspring, the genetic data say almost always the offspring are more robust than the populations that form the hybrids.
One of the questions ask, have there been any environmental studies on the impacts to wildlife that eat these mosquitoes, or that eat any mosquitoes?
The EPA documents refer to a couple of studies by Oxitec. I think one was using guppies, and there was another predator species. They've not been published, and there are no studies on mammals at all referenced in the EPA documents that I've read. So very limited studies. And I did share that quote in my talk that the EPA accepts that there hasn't been enough data show whether or not there are any toxic or allergenic effects, because enough data just hasn't been provided yet.
Okay. Have a question. I think this is for either Dr. Powell or Helen. What percentage of the mosquitoes that are being released are female, and how many will be released? I think you covered that, but try it again.
Sorry, do you want me?
Yeah, either you or Dr. Powell. What percentage?
You had the numbers, Helen, so why don't you, the proposed numbers to release?
Yeah, actually, I don't have the females for this new second generation one. So for the first generation, we've got the data from so Oxitec's always had adult traps as well as the ovi traps, the egg traps that were referred to, but it's never published the data, or only a small amount of data from males. So obviously what we really want to know is how many biting females there are out there. The only place we have data was from Cayman Island report, which showed these massive spikes in females at the time of release that so worried the Cayman Island scientists. This is a different version. We don't know if we'll get those spikes with this version. Oxitec says you won't, but there's no more information.
This is a question for Helen, but I think it's also a question for Jack Norris as well. How much would it cost to test for tetracycline in the environment, and why hasn't it been done already?
I don't know the costs.
All right. As far as the studies the doctors went, it's basically a throat swab type of thing. It's going to cost a couple $100 maybe to go ahead and take a few of the adult tube-release mosquitoes, rub them on some agar, and see what grows. As far as the outside, I cannot comment further on that.
Okay, another question for you, Dr. Norris. Could the bacteria on these mosquitoes develop resistance to other common antibiotics because they're being exposed to tetracycline?
There's a concept called a plasmid. As it stands right now, there's not a lot of research in being done in the bacteria of mosquitoes, what they carry on their surface. I showed the picture of the person standing with his hand out, the cartoon with the bacteria on it. There is the concern that you have is that by going ahead and propagating one type of resistance, oftentimes in the hospital and in the healthcare setting, we end up propagating more than one type. And in this concern of staph, which is very concerning in the Keys, MRSA in particular, there are two antibiotics that are on antibiograms still have great kill. One is Bactrim, a sulfa drug, and the other one is tetracycline. The other drugs don't have as good a strength against our staph as these two. Well, if you're allergic to sulfa, what are you going to do? So I kind of expanded your question a little bit.
Tell people what a plasmid is.
All right, basically there's DNA that can be exchanged between different bacteria, not just in the same species, but different species of the bacteria, and they can transmit genetic code. So if there is think of it like a toolbox, that if you know that you are going to need, you go into the beach, you need beach stuff. Bottom line is those bacteria that go to the beach have their toolkit. Well, in this case, in a bath of tetracycline, where the ocean is full of tetracycline, they need to be resistant or they just die. And if mom needs enough antibiotic to soak into every cell in her body to reach to a DNA switch in her genes, in the, we got the cell membrane, we got the goo, and we got the DNA that makes the baby. And, if it has to get in there, that's a lot of tetracycline. So you can bring an awful lot of things in a toolbox that can be harmful to those of us who get sick with those bacteria. So we ask the question, test it, please.
Okay, this question, I actually think it's for either Dr. Norris, Dr. Powell, or Dr. Wallace. What are more ecological and more effective ways to address mosquito-borne diseases like dengue?
Who wants to go first? Ladies?
Okay, there's no magic bullet. So we don't have oh, here's an alternative that works perfectly, but there are lots of other things that are being researched. There are obviously existing methods, particularly tackling the breeding sites, which can be effective. Just the fact you're living in a wealthier country with screens and so on your doors probably makes it more effective than in poorer countries. But you also need, more research in a wide variety of different fields, including vaccines, including containers that prevent the mosquitoes from breeding. There's a really wide range of options that are under research now.
Dr. Powell?
Aedes aegypti in particular is very closely associated with human habitats, and it's by far the vast majority of its breeding is being done in human containers, like tin cans in your backyards, in your bird baths, and in tires, used tires that have accumulated water in them. And so a good way, and it's been proven to be very effective, is for people to dump stagnant water on their property. That's the easiest, cheapest, and most environmentally safe thing you can do. So.
Okay, Dr. Porter in the Keys, a little Keys history for you guys, 1800s. It was a physician who led the driving out of yellow fever, a mosquito-borne illness from our Keys. The way Dr. Porter did it was he didn't have helicopters, he didn't have planes, he didn't have larvicide, he didn't have mallet, he didn't have any of these things. What they did was they, as the other two speakers said, is dump the water. They went, they cleaned up debris that was all over. As a matter of fact, it was so bad at one point, just prior to the Civil War, that the Union Army almost left the Keys because they had too many sick people. So Dr. Porter later became the first Surgeon General, or the equivalent of Surgeon General of the state of Florida. So that's a little Key West history for you, where one of our docs was the first Surgeon General, just because of things like driving out mosquito-borne illness without helicopters.
Well, one of the one of the questions was, there was a question about liability. What does the company Oxitec have if its trial, what liability does Oxitec have if its trial has negative impacts on the people here or the environment? I haven't given Ed Russo any questions yet. Ed, why don't you start with that one if you want to?
But in terms of public administration, the actual government function in all this is to normally require performance and maintenance bonds put up prior to any of this happening. I would hope that the Mosquito Control Board asks for a significant amount of bond protection. Because, listen, we've just been through Irma and then COVID and then the economic impact of COVID. And now we're going to, just when you thought it was safe to go outside, we're going to be hit with 500 million mosquitoes being released. I mean, what is wrong with these people? But if they're going to do that, you have to realize that what do you think is going to happen to our economy if just one person gets sick and there's an unknown reason for it? You know how the media plays up these things. A couple of days after the release of these mosquitoes and somebody comes down with a rare thing, what do you think is going to happen to our economy then? This is just not needed. Again, it's an economic debacle. It's not effective. And we should be rethinking this and respond to Dr. Norris's concerns and Dr. Wallace's concerns and Dr. Powell's concerns and Milo Demir's concerns. It's amazing. If they're listening to Dr. Wallace and Dr. Powell, how right Milo Demir was. And as you know, she was in the process of delivering a petition of over 200,000 signatures to the EPA against this. She had an untimely accident. And that's just terrible for all of us. We miss her greatly. But to be honest with you, yes, there has to be an enormous amount of protection. Bonding is the way to go. If they move forward on this, has to be significant performance and maintenance bonds put in place. Thank you.
Well, thank you. We're at about 15 minutes after the hour. I do want to tell you all that we have on the Center for Food Safety website, we are live streaming this now on our Facebook site, excuse me. And so you'll be able to go and watch it all over again if you want. Several of you have asked where you could get Dr. Powell's paper. He did post that in his talk. Might ask Dr. Powell once again to just speak it for folks that could take quick notes. Otherwise, we'll also post it on the Center for Food Safety website too.
I think it's just as easy for you to post it. So rather than it's scientific, I forget the volume number and the page numbers. So
okay.
You can post it and you can get at it.
All right. Well, please go to our website and at centerforfoodsafety.org. And Helen Wallace will probably post it to hers at GeneWatch UK, and we'll try to get it around. Let's see. I'm trying to make sure I've got as many of your questions as we have time for. Well, I'll open it to the panelists. Is there anything that you didn't get asked that you should have been asked? Okay. I will there are a couple of questions I felt you dealt with, but maybe it's worth trying again. A number of people thought that these new male mosquitoes are supposed to breed with the wild mosquitoes, and then the offspring will die. Is that what's supposed to happen?
With the older version, so not the one they're proposed to release now, all the offspring were supposed to die at the larval stage, but you had a 3 to 4% survival rate, which is what caused the spread of the genes into the wider population that Professor Powell talked about. This new version, only the female offspring are supposed to die at the larval stage. So the male will mate with a female, it'll lay eggs, they'll all have the genes in, the genetic engineered changes in, and then only those female offspring will die, and the males will carry on for multiple generations, spreading in the environment.
But I mean, JD, the problem here is that we're all talking about testing. This is all about testing. And our feel is that, great, you want to test it? Go test it on volunteers. Go test it on people who think this is a great idea or are going to make a few bucks on this or whatever it is. Apply this to informed adults. Tell people what's about to happen. I mean, in prisons today, you can't pull this off. I mean, you can't do experiments on people unless they have informed consent. And it's not just to tell them, they have to sign legal documents to allow this to happen. But now, because of our mosquito control board, they think they can release this and make us guinea pigs, make us experiment, make us part of this experiment. And we have no consent about it. That's absolutely nuts. So the important thing is, if they think this is good, if Oxitec is so sure about their technology, then do the things that Dr. Norris wants. Respond to the concerns of Dr. Wallace and Dr. Powell, and apply this in a clinical and biological tests on volunteers, not on the general public.
The question I would ask, that I would like people to ask tomorrow, is why wouldn't I take one of these little egg balls and basically carry it to the trash can and drop it in a plastic bag that I seal? Because without the information about antibiotic resistant bacteria, and if it rains and suddenly 1,000 mosquitoes start flying around males looking for the females, and basically by making the females pregnant, they also transmit their bacteria. And I know one doc called this a sexually transmitted disease. But then the mosquito bites a human. I've seen many mosquito bites that become accidentally infected. Well, again, tetracycline is one of our 2 antibiotics orally to treat these things. The question would be, why would I let this thing sit next to my house that is basically not doing anything to study disease management, that has this data floor to it, and so forth? So I would ask people to state, why shouldn't I just throw this thing
out? What you're saying is that the people that get these boxes on their property should ask, why shouldn't I just throw it out?
Yeah, ask your physician.
Okay. Another question kind of related to that, will there be third-party oversight in the Keys that will be implemented to monitor this experiment? And can residents opt out of this experiment, or will we have to move somewhere else if we oppose the experiment?
Wow, what a question. We have been calling for third-party oversight. We've been looking for a citizen review board. You have to understand that Texas and Florida has a sovereign immunity, which means it's very difficult to sue the government. It just doesn't happen. The cards are stacked against anybody that tries to protect his family or his property from government abuse. So it's important that our citizen oversight is aggressive instead of being a rubber stamp or a bunch of cheerleaders. We need our mosquito control board to hold everybody accountable. And if they're not going to do it, yes, there should be a third-party group of specialists or citizens to oversee everything that's going on. That would be a smart move. That would be a very smart move for our mosquito control board to do. Thank you.
Okay, you've got a question following up on that, just to make sure that the current proposal is that the mosquito control board would exercise its authority to put these boxes on private property, and that they have the right to do that. Is that correct?
That's correct. The power of the Mosquito Control Board is one of our first questions, which was used. Again, there were just fundamental questions from the city of Key West back in April 14th, 2012, which is, what's your regulatory authority to do this? What power do you have to experiment on private citizens? And they've never really answered that question. So if they're going to be able to have all the authority they think they have, they better be able to prove it, because even though they have sovereign immunity, people are going to be angry and aggravated by their feeling that they can do anything they want without oversight at all. And that's just wrong. So yes, they have the authority because of their overall mission statement to kill mosquitoes. They believe they have the ability to go anywhere and do anything they want, and even use chemicals that are not approved by the state of Florida's Department of Agriculture, like Vectobac. But that's for
unfortunately, I am getting the signal that we have pretty much used up our time. We've had at one point over 120 of us online, and we're starting to wander away, and that's always a good sign that the crowd is ready to go home and get dinner. So thank you all. I know if we could applaud, there would be 100 folks clapping, and we appreciate Helen Wallace staying up really late, and Dr. Powell and Ed Russo and Dr. Norris and Dana Pearls and Barry Rage trying to keep me focused, and I appreciate all your time, and please go to our websites and go to the Center for Food Safety Facebook site, share it with your friends, and again, thank you all. We will, if you join any of our organizations, we will be sending out more action alerts about what you can do about this, and suggest more things that you might ask the Florida Keys mosquito folks. And anyway, so thank you all for a good night. And one last thing, someone asked, if you've had a lawsuit, why doesn't that just stop it? Unfortunately, the way endangered species lawsuits work is they don't get to put a stop on things, at least not right away. So thank you all. And again, thank
Real quickly,
our
JD,
speakers.
just as a point of information, a lot of questions have been asked that we haven't been able to address. So we will be addressing those and sending out to the entire group with the questions and answers to those questions.
All right, thank you.
Thank you, Barry.
Thank you.
Thank you, JD. Thank you, everybody.
Thank you.
Thank you.